Let me see. So this button. Okay. All right. It appears that everything's working fine. Hi, I'm April Pease. I'm from Texas. I'm going to talk about biophotons as a PhD project. So I finished a master's in physics, and now I have about 1.5 years into a biology PhD, having not had any biology experience. So this is a preview to what my entire PhD is. Okay, so getting the light right. Determining the effects of stressors on patterns of complexity during plant and animal development using biophotons. And there's just a general outline we're going to go through. And let's start with some fun questions. What can we learn about developing organisms at the macro and micro scales by using complexity measurements of biophoton emissions? So thankfully, you guys have been given a preview about what biophotons are, so I don't have to do anything extra. And it turns out all living organisms emit biophotons. So if you're not aware of what they are, everyone has them. A quick quote from Master Yoda, "Luminous beings are we, because we all have biophotons." Okay, so what are biophotons? They are in the 800 to 200 nanometer range, so infrared all the way through UV. They're very low intensity, so you're not going to see them, but they're thought to be metabolically derived. We're going to see some, hopefully, more about the origins through the PhD that I'll be working with Dr. Hunt van Herbing. They are directly generated by living organisms. By that, I mean they are not bioluminescence, they're not glow, they are not phosphorescence, so they're not absorption and re-emission, and they are not thermal radiation. They don't come from heat. They are just directly created from something that is alive. So what can we learn about biophotons? Here's some other questions. Are biophotons incidental products of metabolism? My PhD advisor wonders, if they come from cellular replication, DNA replication. Could they also be from metabolism itself? Do they indicate health or stress? This is a common topic of research for biophotons. And then have they been co-opted for something more? There's a lot of talk about quantum mechanics, but I'm not going to address these questions. These are just things for you to think about. The actual background and interpretation of some of the literature on biophotons. So I'll start with Benfatto et al. This is actually the image from the paper from my PhD advisor where I drew the little seed germinating. Because when I saw the paper by Benfatto, I actually took the average time for a lentil to germinate, looked on YouTube, found that it tends to be around the time right in the middle there between regions 3 and 4, where the seed has the root start to emerge. So a little bit of thing called a radicle pokes out at that point. So that was just from being observant on YouTube. So here's the interpretation. You've got 2 scaling processes. You have renewal process characterized by crucial events in the first stage. This is the part where Dr. Hunt van Herbing is thinking the DNA replication occurs. And then you have the stationary process characterized by fractional Brownian motion afterwards. And so we don't know for sure, obviously, this is going to be the topic of my PhD. But the point is that this may be an indication that there's a type of complexity prior to the sprouting of the seed, and a different type of complexity after. And at the border of these is a developmental marker. In this case of the seed is where the radical emerges. What will be interesting is to look at other developmental markers, because that is not the end of development. Unfortunately, Benfado's experiment only ran for 70 hours, 80 hours. So we need to go further than that. So the point would be to look at much later stages of development and see, are there new complexity changes at those other developmental markers? So as a significance, the fractional Brownian motion type of complexity and the renewal complexity can actually have the same scaling. So in this case, they do not. They do have a different type of scaling, but conceivably they could have the same scaling, which has been a question in the literature for a while. And because of that, people often misconstrue these different types of complexity as the same thing, but they are not. And so Benfado's paper is actually empirical evidence that supports distinguishing between the renewal complexity from a dynamical origin, and the one that is characterized by what Paolo Grigorini's group calls crucial events, and that of the FBM, fractional Brownian motion complexity, which has a stationary origin and is characterized not by crucial events. Okay, so we've got that. And so here's some math. Everyone likes math. So we have the renewal complexity characterized by crucial events. So we describe it by 1 over tau to the mu, where mu is between 1 and 3. When mu is between 1 and 3, the scaling parameter eta is n plus 1. When it's between 1 and 2. When it's between 2 and 3, the scaling parameter comes from mu equals 1 plus 1 over eta. Okay, and so there's a place where things get confusing. When the scaling parameter is 0.5, mu could either be 1.5 or it could be greater than 3. Both of those things are typically not interesting when you're talking about complexity, so we don't really need to go into it. But because of this, there's just some caveats. Okay, so here's that graph again. We're just going to zoom in and look at his paper a little more closely when he had the stripes and what those scaling values are. And as you do that, you can see those asterisks. So just as the point, the mu value might not actually make sense for those non-renewal processes that occur after that seed radical emerges. Okay, so let's go on to some other research. So if you don't know a lot about biophotons, you might be curious what else there is. And so the most interesting one to me is actually from the Kobayashi lab in Japan, where a person was smoking a cigarette, and you can see the region they were holding the cigarette. So this is where a lot of biomedical research focuses on stress. So clearly, if you know what's in a cigarette, you know why there is stress in that area. Another one was from a name in which I cannot pronounce, but it was an infected plant that was then inoculated. And what's interesting is this little peak is after it was inoculated, there was a change of the biophoton emissions. So you're going to start to wonder, what is stress? What are stressors? What is health? Because that's a common thing that people talk about when they talk about biophotons in medical research. Okay, so let's talk about stressors, stress and vitality. Suppose that vitality is an increase in biophoton emission above whatever the baseline is due to a stressor. A stressor being something that could be good, could be bad. It's just a thing that makes a change from the baseline. And then suppose stress is a decrease of biophoton emission below a baseline due to a stressor. So in this case, we're thinking not healthy. So here's an example for you. If you have exercise as a stressful thing that occurs to your body, but improves the overall health of your body, yet you have overtraining where it decreases your overall health. So you can start to think about what is stress, what is vitality, what does a stressor do, and does it change the amount of biophoton emission? So let's go towards some underlying biological mechanisms. So for my project, we want to find out some biological mechanisms using lentil seeds, but also compare them to zebrafish, which is a model organism, and it's really great for NIH funding and stuff. But it's possible that biophoton emissions can be a reliable and quantitative measure of health and disease. So that would be a good thing to look for. So just as an idea of the methodology, so I said that we're looking at this seed radical emerging as one developmental marker, but there are others. So the idea would be measure the biophoton emissions for the duration of several markers in the lentil and the zebrafish. For instance, there's shoots and roots in a plant, so there are obviously more developmental markers that we need to look at. In the zebrafish, it's actually a little different because it's an animal. Seeds in animals are very different. So for a lentil, the development of where fertilization occurs in the parent plant, but we are looking at the germination of the seed well into development. But for the zebrafish, obviously, post-fertilization is very easy to see. You have the fertilization event, the cell divides, a bunch of time occurs, and we know a lot about the development of zebrafish. They are invisible, so you can see through the eggs. And so you can watch as it develops. You can see at each of these developmental stages, and they're characterized by different timings. You can see that at specific times, these things occur, and the timing actually changes. So you have an event where, okay, lots of rapid cell division, new marker, the timing changes. And so we could correlate those to these developmental markers to biophoton emission. This would be what we're planning to do. The other thing is to compare the effects of different stressors like temperature and pH for lentils and zebrafish, and then for the lentils, moisture and soil characteristics. There's a lot of things there that we can do to characterize stress versus health. So let's take a little detour about characterizing health. How do we even know the seeds are stressed? So you imagine just a petri dish, you throw some cotton and some water and you start germinating. Is that seed happy? I don't know. So I started thinking about that. What are optimal germination conditions? I'm not a biologist yet. I've only had a little bit of work with the biology thing, and so now I'm starting to think about these big questions. There's this place called Italy you might be familiar with. Dallas got one. And so I grabbed some lentils that were from Italy. And I was like, okay, well, these might be what they were using. And are they special from Italy? I don't know. And yeah, apparently lentil seeds from Italy are special. It turns out they prefer the small seed varieties. Everyone else in the world likes large lentils, but Italians, they like them small. And so there's this lovely region in Umbria where my seeds came from that spot on the left, the arrow. The Castelluccio is the famous one with the beautiful flower fields, and that's somewhere I'm hoping to visit soon and see what the difference is. But one of the differences is, we'll go into in a moment, but there's this paper, Cash et al. 2001, who said a few things, just how do you make lentils, right? So, whoops. So it turns out they are drought tolerant and they're a cool season crop. I think of Italy, I don't think about cold, snowy, mountainous places, but that's where lentils like. And they're actually planted quite deep, 5 centimeters, which means they don't get much sunlight in the ground. The first leaves form below the soil during germination, so those developmental markers actually will occur in the dark, which is great for biophoton research. And it's actually great for frost resistance. That's part of why they like those mountain areas. And it turns out that if there's too much nitrate, it's not good for them. If there's not enough phosphorus, it's not good for them either. And all of that will prevent the nitrogen fixation because it turns out lentils are able to take a host bacteria and use it to extract nitrogen from the air, which is pretty cool. And then weed management prior to lentil establishment is critical. This is the general commercial crops suggest weed management. However, you're going to find out, if you're from Umbria, that is not the case. So, this is the beautiful Castelluccio, and I'm wondering, well, how do I make stress-free seeds? These look like they should be pretty happy. Plenty of flowers around, it's colorful, it must be great. And it turns out that in Castelluccio de Norcia, they're grown among white chamomile, leucanthemum, they look like daisies, wild mustard, Venus myrrh, cornflower, and you get white, purple, blue, and red flowers. It's beautiful. So unlike the rest of the world, they don't mind having weeds around, and they let them all grow together. And so you end up with a very organic thing, and it turns out the USDA food grants, they like to fund interesting projects like this, so comparing the commercial crop to the Umbrian crop might be an interesting way to get funding. So, no pesticides, no weeding, and a mixture of flowers. Here's a quote: local farmers who, to preserve the fields, did not use pesticides, therefore maintaining organic cultivation, during the times when lentils are cultivated, have benefited to have always let other plants flourish in the land, gaining many benefits. In this way, while waiting for the lentil to grow, weeds have taken over, recreating a natural flower show with plant species in different colors. So lots of Italian journalists like to go here and take pretty pictures, and I want to do that too. But aside from that, there's some interesting questions about this soil, and I'm hoping I can figure that out. Because if you have lentils and you're trying to get this great pH, but you've got nitrogen fixation, so it's changing the pH, but then you have all these other plants around that are going to change that. So how does that work? And what are the microbes in the soil doing? And what is the host doing? So there's all sorts of great questions to ask that are related to biology and environmental science. All right, so here's the questions. Is there something special in the soil beyond the karstiology? I found out what karstiology is. It's very interesting. Are there differences in the microbial abundances? Is the lentil symbiont different? Because I found a cool paper in India that said, while it's the same variety, it has a different symbiont. Are the seeds happier? They should be. I feel like that's a nice place to live. And are bigger commercial lentil varieties stressed? Because if we're going to make a sustainable world, maybe we need to learn how to farm better. And I don't think commercial farming is doing a great job at the moment. Maybe we can help them out. So that was the end of the detour. So let's just make some quick ideas for experiments. I just have some ideas of adding some soil and adding lentils, taking out the soil, just doing the lentils, doing both together. Because imagine you have biophoton emission from the soil because there's all these microbes in the soil. So you want to measure what that is. Then, of course, the soil plus the lentils, see how they work together. Maybe there's an increase beyond what you expect of just additive. And then there could be the same method that Maurizio did. Maybe that's really what's fine. Maybe there's no difference. So I want to test all those things. And of course, in biology, you have to do lots of replicates. And in tandem, I'd like to do some germination for picture purposes because we don't actually have any about when those stages occurred. And so I kind of had to make the hypothesis from the YouTube video. So having the germination pictures would be great. And in the meantime, I'm going to head some wonderful places in Italy. I'm very excited. And set up a LabVIEW program, work on figuring out some experiments, and then make a few really quick results, hopefully. Bring that knowledge back to Denton, make some setups, and hopefully prepare for next summer where I can come back and we can do some genetic sequencing of the soil and see what's in the soil. And then start coming up with my zebrafish plan so I can compare the two, build some new setups, and do some genetic sequencing of those microbes because your gut and your soil are actually pretty similar. And there's a lot of different things to do. And So the very next steps, just biophotons have had implications for non-invasive imaging of stress, and that's pretty cool. However, their origin has been disputed. So maybe we can see, is this mitogenic radiation? Maybe it's from cellular and DNA, well, DNA replication. And then we can see, you know, maybe there are new ways to characterize biological processes. If I know the exact time of emission at one developmental marker, maybe I can change those tables that are a little confusing because their timing doesn't make sense to me. And so I'll work with Maurizio and learn some more developmental biology, add in some complexity, and start looking at the health. And that's about it. Any questions?
Thank you very much, April. There are questions? Question?
Thank you for your talk. I have more curiosity than proper question. You said you are going to sequence all the microbiome.
It was 16S, yes.
Which kind of sequestration method will you use? And next generation sequencing or amplicon sequencing method?
So I don't know exactly, but I've been told 16S is what I'm going to use.
It's amplicon.
Ione knows.
Yeah, it's amplicon. So it's 16S using amplicons.
16S using
Amplicons. Amplicons. Sorry, can you hear me? Amplicons.
Okay, we don't understand that language.
Amplicon, I took away from this talk.
Thank you, Bruce.
The question is, why are you sequencing the microbiome associated to your model organisms? Are you thinking that there is a correlation between biophotons emitted by lens or zebrafish and the bacteria they can host? There is
So there
possible
might
relationship?
be, but specifically they can be related to stress as an indicator of stress. So if you have something wrong with your gut, your entire body feels it. And if something's wrong in the soil, an organism's going to feel it too. So it's useful to know what's there so that you can see as a characteristic of the soil what is healthy, because if you can associate stress or health with that biophoton signal, it's good. And that's one way you could do it.